Hey ladies, welcome back to my channel. This is being ridiculous here. All right. So, in our previous lesson, we spoke about organelles and I gave you a quick introduction to cells, why they're important, how we view them with microscopes, the light microscope, the scanning electron microscope, and the transmission electron microscope. So today, we're going to be looking at the different types of cells and we're going to dig into more so of the bits and pieces of the actual cell. So we're going to look at what they look like. So if you would like to follow as well, you have a version of this on page 118, which doesn't really show like all the sharing bits and pieces, but I mean you can use it in your textbook. Also, if you guys don't have a textbook, you need to let me know so I can start taking pictures of things. Just holler at me. All right, so in front of you, you can see a plant cell and you can see an animal cell. So on the right-hand side, you'll see an animal cell. On the left, you'll see a plant cell. Please forgive the fact that these are terrible diagrams. There's lines crossing all over the place, but it's a nice way of you to visualize this whole thing in 3D. All right, let's have a look at the differences. So they've made them pink and green, which is absolutely atrocious as well. But they're not really that color. They're actually colorless, and I'll show you in the picture just after this. You can see that they both have a nucleus, and that nucleus is a big round purple ball in both instances, and right in the center there is something called the nucleolus, which is really what we're going to be focusing in on this lesson. It's just the nucleus, and a little bit of the living components. So over there, I don't expect you to be that interested in the nuclear pore, nuclear envelope, nucleolus, and chromatin just yet. I do want to point out some other things, that they both have a mitochondrion. The plant cell has a huge vacuole, whereas the animal cell has like a very minimalized, and to a large extent they just don't have a vacuole whatsoever, which you should have learned last year. Then both of them have ribosomes, both have a Golgi apparatus, both have this thing called endoplasmic reticulum, which we'll get into a little bit later. They're smooth and rough, or SER and RER, and we'll go over how we indicate that later. Notice that they both have cytoplasm, which is obviously a given, otherwise we spoke about it earlier. And then things get a bit pear-shaped where we look at where it says cell membrane and plasma membrane. Those are the same thing. It's just different names for exactly the same thing. And the bottom up, there's a little key where it says like there's a double star or a single star, so on and so forth. The other thing the plant cell has is a chloroplast. So those are beautiful little green things which make them will allow them to go through photosynthesis. And on the animal side, they're the only ones with centrioles, which will be quite important in the next section for mitosis. You can also note that they have these little vesicles. You'll see the word every now and again. There's one called secretory vesicle on the animal cell in the bottom right hand corner, and we'll go over what those are in like probability. So later on I'm going to expect you to be able to draw a table off by heart, and for your exams where you have to be able to compare an animal and plant cell at all times. Okay, so let's have a look at what these things look like under the microscope. So you have the animal cell over there, and you can notice that it doesn't have a defined structure. So we can use the word amorphous. So it doesn't, well, morph means structure, and a something is opposite. So it doesn't really have a structure. It's more like just this kind of round-ish structure, but you do see that black little spot that they were talking about earlier, and that thing specifically is a nucleus. So what you're looking at over there is someone has like scraped the inside of their cheek and they put it on a slide and they've stained it with some fuchsia red, and that's what you're able to actually see. In the bottom left hand corner, you'll see the plant cells, and there is a far more defined shape. So it's more rigid, more tightly packed as well. So you notice that they're not all randomly floating about. This is probably from an onion cell. So a lot of the times you can peel off like a piece, like if you snap the onion in half and you peel off that thin film, you can actually look at that under the microscope. We can do that when we get back to school. But you can also see the dark spot in there, which means that they also have a nucleus. Like, it is extremely important that everything has a nucleus. There will be instances where some cells don't have a nucleus, and we'll get to that a little bit later. Especially when we do animal tissues, you'll notice that a red blood cell does not have a nucleus, and we'll go over that a little bit later. Okay, so we got components of the cell. This is just building up. And I also want you to refer back to your notes as well and have a look at all these different like organelles. So on your page 118, if you can just like write down all this information from the slides onto there. So I recommend you draw a big version of those and then add this information. It'll be a good way to study all of this. So the living part of the cell, and there's living and there's non-living. So the living is called the protoplasm. Proto meaning like before, plasm meaning liquid. So, and that includes your cytoplasm as well as the nucleus, and these are the living parts of the cell. So I've included a little beautiful diagram over there. You can see the diagram over here, and well, the pink cell, and it says there's cytoplasm plus nucleus equals protoplasm. This is important, and I like to ask this in multiple choice, so it's a good idea to like just fully understand what that means. Also, the term cyto meaning cell. So cytoplasm, liquid of the cell. And remember that cytoplasm, which we'll get to later, is majority made up of liquid, specifically liquid water, and that is because of all reactions can take place in there, we can move things in it, so on and so forth. And then we have the non-living components, which is only in plant cells, and they have this gorgeous cell wall. So you would have noticed from the previous slide, the piece that was green was notably like thicker and had this really big structure, and that was a cell wall. And if you remember from our previous section of work, a cell wall is made out of cellulose. Okay, so let's have a look at the nucleus itself. So the nucleus, there is a nucleus in every single cell. And even if like it was an original cell, like the original cell itself, even if it lost its nucleus, it had to have had a nucleus as well, because it needs to tell the cell what to do, when to do it, how to do it. So the instructions come from there, and that's why a lot of the time it's called the brain of the cell. So it's made out of three main parts, and this is the nuclear membrane, nucleoplasm, and chromatin network, and then finally the nucleolus. So what you'll notice is there's another membrane inside of the cell membrane covering this whole thing. And you can imagine that it's extremely important to have a membrane around this chromatin network, which is actually the DNA, because you don't want any random chemicals that are floating around to just have access to that DNA, which could ultimately change it, cause mutations, and so on and so forth. And obviously that nuclear membrane needs to be extremely semi-permeable, so it needs to be able to only let certain things in and out. And you'll see later when we actually draw it that it's like a double membrane, which really causes a lot of things to change. And then finally the nucleolus, which is really important and not something we often think about. So this is just a 3D version of the whole thing. So you'll be able to see that the nucleus itself has this 3D more rounded structure. It's surrounded by the nuclear envelope or nuclear membrane. In the nuclear membrane itself are nuclear pores. And these nuclear pores are extremely specific in what they allow in and out. So I may have mentioned extremely briefly when we did proteins that you can have something called a transport protein. And the transport protein is what you're going to find in those nuclear pores. Those nuclear pores, like substances or like molecules, actually bind to the pore themselves, and then the pore decides whether it wants to let it in or out, either from inside the nucleus out or from the cytoplasm into the nucleus. And please remember at all times that there's cytoplasm around each one of these organelles, so don't just think these things are randomly floating around. Then we have the nucleolus itself. The nucleolus is known as the dark spot. Now please don't get confused, because in previous slides in the introduction I mentioned that scientists saw a dark spot. That dark spot was the nucleus. But now inside of the nucleus, you find the nucleolus, and that is also known as the dark spot, and we'll get to what that is a little bit later. Finally, we have something called chromatin and chromosomes. We'll get to the difference between those a little bit later, but please just know that chromatin and chromosomes are both made out of DNA. It's just in what shape and in what structure, and the reason for that shape or structure is dependent on what the cell wants to do. So if it wants to go through the process of mitosis, it wants them in the shape of a chromosome. If it wants them for daily functions, it wants them in chromatin, and I've got some pictures later on to show you. Okay, cool. So I do expect you guys to write this down and just make sure that you are fully aware of all of this. So we got the nuclear membrane. It's a double membrane. It is important to recognize that it is a double membrane. So there's a space between the cytoplasm and the inside of the nucleus itself. So there's a membrane, some space, and a membrane again on the other side. And that is really important to stop things from being able to get through, because you can imagine diffusion can also just take place. So you want a double layer just to stop everything going around with the nuclear pores. And the pores control entry and exit of substances. So when you write that down, please mention there that the pores are made out of proteins, and these proteins are extremely semi-permeable. Like, if it's not meant to be in there, it will not be in there. Then we have the nucleoplasm and chromatin network. Technically, these should be split up, but we'll keep them as one, because it kind of makes the entire interior of the nucleus. Sorry. So, the nucleoplasm as well. So, nuclea or nucleo means of the nucleus. And then plasm means liquid. So, this is nucleoplasm, the liquid of the nucleus. It is gel-like to hold all the structure inside. Now, chromatin network is really important. This is one of the first instances where we now hear of DNA. And we don't just call it DNA, because DNA, I mean, we all know it's made out of DNA, but what shape it's in is more important. Chromatin network is more of an unwound version of a chromosome. So a chromosome is like really tightly packed together, whereas chromatin network is a lot more open and things have access to it a lot easier. So the chromatin network is found in the nucleoplasm. It's made up of DNA or deoxyribose nucleic acid. It carries all genetic information. You'll recognize that containing or carries all genetic information is the same biological importance that you found when we were doing the nucleic acids in the previous section of work. So go back and have a look at it. So this genetic information is a set of genes which are transferred from mom and dad to you. The cells divide during mitosis, and each new cell receives a complete set of chromosomes. Okay, so what you'll recognize is that you get half of your DNA from mom and half of your DNA from dad. So you get one set from mom and one set from dad. And this creates a complete set of chromosomes. I expect you to go and look at how many chromosomes you actually have in total from mom and dad. If it's anything less than 40, you're wrong. And if it's anything more than 50, you're also wrong. So during mitosis, chromatin network thickens and becomes chromosomes. And we'll deal with how that thickening and winding up of the chromosomes occurs a little bit later. It's not that necessary now. You just need to know that the nucleoplasm is gel liquid and the chromatin network is DNA, and it's an unwound version surrounded by a nuclear membrane, which is double and has extremely sensitive, semi-permeable nuclear pores. All right, this bad boy called the nucleolus is the dark area within the nucleus, and I'll show you when we're drawing it a little bit later what I mean. And when you draw a diagram for me, often you're going to want to color this in because all diagrams have it. And if you don't draw it properly, you think that you've drawn a nucleus in a cell. So it starts to look very weird very quickly. But I'll guide you through that a little bit later. So some cells may have one and others may have many. It's made up of RNA and protein. So now let's quickly just remind ourselves that RNA can be made from DNA, and then protein can be read off of RNA and made as well. So now's the idea that all DNA carries these genes, which is a short piece of DNA which codes for specific characteristics. You're going to use that definition a little bit later, so I'll say it again. A gene is a short piece of DNA or short segment of DNA that codes for very specific characteristics. So the DNA holds the code, RNA is made off of that code, and then the RNA is read by a ribosome, and that is what makes protein. So this is protein synthesis, which only arrives in matrix, so we're not going to go into that, into the nitty-gritties of that. And then finally, it's involved in protein synthesis, which I kind of explained already. Functions of the nucleus, importante. So, it controls all cell activities, which means that if you do not have a nucleus, it means that you will not function as a cell. And if your nucleus becomes broken or ripped apart for some reason, it means that the cell will actually just cause self-suicide, which I mentioned earlier, called apoptosis. Finally, second last, it's involved in cell division, known as meiosis and mitosis. So I'm not going to give you proper definitions for those just yet, but ultimately mitosis is the division of one parent cell into two genetically identical daughter cells. Okay, so you take one, you copy it, you make two clones, and they are absolutely identical. Whereas meiosis, it's for the formation of gametes or sex cells, so eggs and sperm. Whereas you take one parent cell and you make four genetically unidentical cells. Okay, and that is important for later. Whereas mitosis, you get one full set of chromosomes. In meiosis, you get only one set of the chromosomes originally, but that's for another day. Finally, it contains DNA arranged in genes, and this controls all hereditary characteristics. And think of hereditary as the term of like, you got it from your mama, or you got it from your dad. I don't know what you call your dad. It's got awkward over there. So anyway, hereditary characteristics will be like hair color, height. I'm trying to think of other ones. Eye color, skin tone, the shape of your nose, the shape of your mouth, the shape of your face, how much hair you have, how little hair you have, and all those other bits and pieces, which kind of sets you up to become who you are as a human being. So this is just a lovely like 3D version of the whole nucleus as well, so that you can have a good look. It is the nuclear envelope on the outside as well, which is a double membrane. So you can see at the top and bottom there's a layer, and then there is a gap in between them. There's nuclear pores on the right hand side, and just above that I've also included a picture where you can visualize how this nucleus sits inside of these cells. So don't think that they're randomly floating around. They have a very specific spot. A question that you can think to yourselves is, does each cell have a very specific place for the nucleus? And I want you to think about it in terms of like, would you want the nucleus that's in your skin or in a skin cell to be in the same place that you want a nucleus in a cell in maybe like the lining of your stomach where there's lots of acid? Think about where the nucleus would be in the cells in your intestines. Now they look like little pillars. So do you want them, like if all the food's coming through here, do you want like the nucleus right at the center so they can be affected, or do you want them at the bottom? So don't just think that the nucleus does all these bits and pieces. It also is in a very specific area, so it can do a very specific job. And that will come up as we go through the next section of work, which is animal and plant tissues. Finally, you have the nucleolus over there. And the one thing that they've left out of this diagram specifically is chromatin network. This chromatin network itself is extremely important, and it surrounds that entire thing. But let's have a look at what the actual 3D structure looks like. So over here, you can see that there's purple and blue. Now, I don't want you to be fazed by the fact that there's purple and blue. What I want you to recognize is if you look in the top right-hand corner, you can actually see that this is forming that double helix structure I was talking about in the previous section of work. So you can see that it's like a helix, but it's running all the way around. And it's not wound up in a very particular way that it doesn't have a specific shape. It's kind of all just free floating and allowed to maneuver around. When I want this chromatin network, which is made out of DNA, to do a very specific job, however, I then, oh man, I went back the wrong way. All right, well, it was the chromosome. So maybe you can pause and go back and have a look at what the chromosomes look like. On that particular slide, I want you to count the number of chromosomes themselves. And from that actual chromosomes, I want you to figure out whether that is a boy or a girl. So you're either going to look for XX, which is a woman, or XY, which is a guy. And I want you to look at like the actual structures and how they create this like X shape. And that is what a chromosome looks like. And it's the previous slide with a chromatin network that is just super wound to create these beautiful little chromosomes. All right. Now, to almost end this lesson, I don't want you to draw this particular one. I've got the next slide. I want you to notice just a couple of things first. So your diagram is going to look extremely similar to this, but what you will notice is that you have this dark spot in the center. It's a little like dark purple circle, and that is called the nucleolus. Remember that it's made out of ribosomes, RNA, and protein. Which just means that ribosomes are inside over there. Then we have the chromatin. This is also known as the chromatin network. So it is an unwound version of DNA, which allows for natural daily sort of metabolisms. Then we have the nucleoplasm, which is the lighter purple version of everything inside over there. We have a nucleopore sort of towards the bottom over there, which is a hole, which has proteins which stop bits and pieces from going in and out as they feel fit. And we have a nuclear envelope, which is a double layer. Finally, we have this Christmas tree, or I don't know, like river version of everything. And you can actually like follow, you'll notice that it's all pretty much one big organelle attached to the nucleus. So I'm going to say it again. It is one big organelle attached to the nucleus itself. And this is important. And this is the endoplasmic reticulum, and then it is right on the outside of the actual nucleus. Now, if you look at the uppermost layer of the nuclear envelope, you can see to a large extent that the endoplasmic reticulum forms off of the chromatin network, which is extremely important. So anything that is produced inside of the nucleus, let's say for instance, the DNA has been converted to RNA and it needs to be read by a ribosome, it moves directly from the nucleus through the nucleopore and then directly hits the endoplasmic reticulum. We'll get to what the endoplasmic reticulum is for a little bit later, but you can notice there's a whole bunch of red spots all over the place, and those red spots are actually ribosomes. And those ribosomes are directly related to protein synthesis or making of proteins. Remember, the word synthesis means to make. So what are we making? Proteins. And this will be known as your rough endoplasmic reticulum. And it's directly made off of the plasma membrane or the nuclear membrane of the nucleus, which is really important. So if anything needs to be made really quickly in terms of protein, it can be created inside of the nucleus and then directly moved into the endoplasmic reticulum. When this happens, it means that we can produce stuff like, what is that bad boy? Adrenaline or cortisol or estrogen or testosterone, all those like really important bits and pieces. If we need it in high amounts very quickly, we can quickly produce the code from the gene on the chromosomes or on the chromatin network and move it directly into that endoplasmic reticulum. Finally, I want you to draw this by yourself. I need you to learn this off by heart, and I've put it like a huge important there as well. So you notice that not all the labels are on here. So what I want you to do is draw this properly as best as you possibly can. I want you to label nuclear membrane. Where it says DNA, please change that to chromatin network. Because it's just, I mean, DNA is very unuseful. Like, you don't know what you're actually looking at. Then we have the nucleolus. If you color this in, I will rip your page up. It's as simple as that. So please note that it is a spot inside of the nucleus, but it's not colored in. We just colored it in this instance so that you can tell the difference between what the nuclear membrane looks like and what the nucleolus is. Finally, we have nucleopores, and I need nucleopores. They're in the same spot. So the way I recommend you draw this whole thing is you draw one circle on the outside, a circle on the inside of that, erase lines through it, and join the edges of those two circles together. That will create the outside. Draw some squiggly lines in the middle, and finally I want you to draw the nucleolus. I also need you to add the nucleoplasm. So once you've done all of this, I recommend you go back into the video or you go into your textbook and you write down the use for each one of those labels directly next to it. Once you've done that, it will give you a good understanding of what's going on. And then I want you to please go to activity 4 on page 112, and I need you to complete that whole little exercise on the nucleus over there. If you have any problems, well, this will be one of the lessons we're going to talk about in class. So I'm going to give you some time, and then you are just going to tell me how to complete it in class, and we can have a discussion over anything you're a bit confused about. All right, so that was a nucleus done. Next, we'll look at the cytoplasm and some other organelles.